Why do so many drugs resemble dopamine? I'm Colin Flanagan. This is my General Chemistry 1 capstone project. So we're going to start out by asking, what is dopamine? And the answer is that dopamine is a powerful neurotransmitter that is involved in creating sensations of pleasure and feelings of reward in the brain. It is important in reinforcing activities that are perceived as rewarding and highly regarded as an important evolutionary trait which allows animals to learn about their environments. Dopamine is released in response to activities that are perceived as rewarding, such as eating food and reproducing, signaling that these actions are important to survival and encouraging animals to repeat these actions. So dopamine is a neurotransmitter, and it doesn't work without receptors. So dopamine binds to dopamine receptors, and relates actions to feelings of pleasure that are important for survival. The binding of molecules to dopamine receptors is what facilitates these sensations of pleasure, and the actions that cause this dopamine to be released are in turn carried out again and again. So here's a little video on how dopamine is released and accepted by these receptors.
Communication between two neurons begins when an electrical impulse called an action potential travels along the axon of a presynaptic neuron toward the axon terminal. The action potential cannot cross the synaptic space. When it reaches the axon terminal, it causes membranous sacs called vesicles to move toward the membrane of the axon terminal. The membrane of the vesicle fuses with the membrane of the axon terminal, enabling the vesicle to release its contents into the synaptic space. The molecules released from the vesicles are chemicals called neurotransmitters. They drift across the synaptic space and bind to special proteins called receptors on the postsynaptic neuron. The binding of a neurotransmitter to its receptor can trigger an action potential in the postsynaptic neuron. That electrical signal then moves toward the cell body of the postsynaptic neuron. Now that the neurotransmitter has relayed its message, it releases from the receptor into the synaptic space. Some of the neurotransmitter is degraded by enzymes in the synaptic space, and some of the neurotransmitter is carried back into the presynaptic neuron through transporter proteins. The neurotransmitter that is taken back up into the presynaptic neuron is then repackaged into vesicles that can be released the next time an action potential reaches the axon terminal. The entire process repeats each time an action potential reaches the axon terminal of the presynaptic neuron.
Okay, so now that we have an understanding of how these neurotransmitters and receptors operate, we can go and look at how certain drugs can take advantage of this system and overload the system with dopamine. So why are drugs addictive? The first reason is that they release high amounts of dopamine when they are ingested, signaling that a pleasurable action has been taken. The brain's neuropathways takes this to mean that an action that is important for survival has been carried out, which then it incentivizes to be carried out again and again. So the issue with drugs is that they release up to 10 times the amount of dopamine as natural sources, such as finding a good source of food or water or reproducing, which overloads the brain's pleasure center. Other actions that were seen as pleasurable before no longer seem that way due to the larger amounts of dopamine released through drug use. Addictive drugs also overload the ventral tegmental area of the brain, which is an important dopamine regulator. This allows high levels of dopamine to be released in the nucleus accumbens, thus enabling a great sense of reward and encouraging further drug use. So here are various molecules compared to the structure of the dopamine molecule. As you can see, the MDMA molecule, methamphetamine molecule, fentanyl molecule, and tetrahydrocannabinol molecule, which is the main psychoactive ingredient in marijuana, all resemble the dopamine molecule very closely. Methamphetamine and fentanyl are considered as two of the most addictive drugs that exist. The molecular structures of these drugs are very similar to that of dopamine, which allows them to bind to and flood dopamine receptors in the brain. This graphic shows how methamphetamine interacts with dopamine receptors. The methamphetamine molecules in green fool the cell into dumping dopamine into the synapse, causing a surge of exhilaration that normally would not have occurred. Now, different drugs also interact with the ventral tegmental area of the brain, as we see here, even if they do not have a similar structure to dopamine. Other drugs can interact with the ventral tegmental area of the brain, even if they do not have a similar molecular structure to dopamine. They can impact dopamine production without directly binding to dopamine receptors in the nucleus accumbens by interacting with the ventral tegmental area of the brain, leading to dopamine production. Drugs can release higher amounts of dopamine than healthy activities, as can be seen in the diagram here. The increased sensations of pleasure that come from doing drugs compared to eating food incentivize taking drugs in the brain's reward system. When cocaine is ingested, the dopamine neurotransmitter and receptor becomes overflowed with dopamine, whereas while eating food, there's only a small amount of dopamine. This prioritizes the cocaine over the food, as there is a larger reward associated with taking the cocaine. Now, how does all of this relate to general chemistry? We have learned over the semester in general chemistry about Lewis structures and how molecules attain their shapes, from the location and number of bonds and lone pairs in their structures. By looking at the Lewis structure of dopamine and comparing it to the Lewis structures of many drugs, it becomes clear why these drugs impact dopamine receptors and lead to substance abuse. The Lewis structures of MDMA, methamphetamine, fentanyl, and tetrahydrocannabinol, shown in the above slides, closely resemble the Lewis structure of the dopamine molecule, allowing these drugs to bind to dopamine receptors and influence behavior.